welcome back. Here's some information about if you're interested in animal science, so maybe veterinary medicine or research and work in animal science. There's going to be an information session from Tufts representative here, in about a month. And if you're interested in that, there's going to be some signs put up, but I can also share this. Then our first exam is 1 week from today. And there's some more, smaller pieces of information, new information that I'd still like to go over today, but we also have to get ready for our exam. And so, you're welcome to ask as many questions as you want today. We don't have class on Tuesday because Monday and Tuesday are fall break. I will be here, and throughout the next week I'll be checking my email. You can send me questions about the exam by email. It's kind of clunky to answer questions by email. We can also set up a time for a Zoom meeting, even during fall break or like that, because I'll be here Monday and Tuesday. Sometimes a student might ask some questions, and I'll realize, everybody's going to want to know the answer to that. So I'll make a little video and I'll post that. So be aware that we might have another announcement or two that comes out to, clarifies, something about the exam. But you always get an email announcement of that, an email for each of those announcements. And that can come up, like that. So, just let me know, and we can answer questions today. Be aware that that the exam is you have to answer the question. So there's that old exam. There's going to be some fill in the blank. What's this? What's that? Then some shorter answer questions. And be aware when I'm asking you the question. Sometimes it doesn't say give examples, and you don't have to give examples. Just define orthologs. But lots of times it might say, define ortholog and give 3 examples, and that's what you have to do. But just be direct in your answers and efficient, because there'll be quite a bit of writing. Then be aware that there could be 1 or 2 20-point questions on the exam, and it would just be a big chunky one, like describe the vertebrate phylotypic stage, and you got to use all those terms and briefly explain each term, like that. Or there could be a different way of asking that. Maybe there'll be a picture with kind of fill in the blanks with arrows, on the exam. And so I've emphasized that it's good to identify the big, chunky questions and get them ready, like you'd get ready to sing a song. Repeat that. You can check yourself. How much do I know about the phylotypic stage? And and add more to it. Like that. And we have other big chunky things like that. We learned about nematodes and genetic screening, a bunch of terms and concepts there. And those could be 5-point questions or bigger questions. And, if I don't ask you about the phylotypic stage in a big question, there'll probably be some fill in the blanks about what's this, what's that, like that. And be aware that the final exam is exactly the same format and exactly the same question. So we're trying to learn this stuff well enough that you'll be able to go back and look at it during the final exam. Then also be aware of how, your best resource for studying for this exam, and definitely for the final exam, will be notes that you've taken, and rather than like looking at the book a bunch more, notes you've taken from the book and things like that. So you're building that resource as part of this recall process. I accidentally left my phone at home today, and in order to get access to Brightspace, I need my phone to, do that double confirmation. And so I was going to add the handout, and I forgot to do that until this morning. And I'll add this handout too as an announcement as well as a recording, but I won't be able to do that until I get home and my phone later today. So that's that. And then, what we're doing this week is asking questions about Chapter 3, because we're going to sort through that. There'll be a few points from chapters 1 and 3. That's our only reading, but it's a bunch of dense reading on the exam. And then, the answer key videos are ongoing learning, and then we have these 2 assignments. One is due today. And the purpose of that building assignment is to do something different. And, you know, some people really click with that kind of learning. And I think it's beneficial for everybody to explore that. You know, we have that capacity here or potential here in this course because biology is really physical. This is all really physical stuff, and you could map it out. You could literally act it out or dance it out, you know, like that. So keep that in mind. And so that's where we're at, and you know, we can go over stuff if you want, or I can talk and add a little bit more stuff as we go forward today. So, do people have any questions?
I guess I don't have a specific question, but I was hoping, because I know you said that we're going to jump into a little bit of chapter 2 stuff today.
No, not chapter 2. We're just not going to get to that, and it wouldn't be on the exam. But yes.
Oh, okay. I guess I was just wishing you would do a general review of maybe what are the big concepts we should know, which I think I know what they are. But
Yep.
just to confirm.
Yeah. Well, so sure, and we can think about that right now, right? And you know, and I can just, you know, I won't challenge you as a class, but what do you know about muscle differentiation? You know, right? We have those videos from the start of the course, right? And then we've had those quizzes, and the first one was about orthologs, paralogs, and alleles, so some gene stuff. And the second one was about the muscle videos. And then the third one was, I think, was it about the nematode and genetic analysis in the nematode? And in the nematode, we had that assignment that's on our electronic reserve from the previous edition of the book to read about nematodes. So there could be a couple points on the exam from that reading as well, separate kind of reading that's not in this current text. And then kind of two parts of the nematode are the biology of both the programmed cell death story and the RNA interference. And there are some short answers to those, you know. And then we did that genetic analysis, and we learned the terms about different mutant alleles and, you know, how they're produced in nature spontaneously, or they could be induced in lab. But importantly, both of those are random. So you have to do these big genetic screens where you look through organisms, either out in nature, you go look and out in, you know, nature to find a defective organism and say, well, let's study this, or the equivalent in lab. And we just got a little bit of taste of what it's like to do a genetic screen, genetic analysis, because we said this first test you do is a complementation assay, you know, to find out how many genes do you have represented, like that. Yep. And I'll just mention, we won't dive into this, but I'll just mention this, that we talked about hypomorphic alleles, loss of functional alleles, and we had this example where, the gene is completely destroyed. It's a null allele. And we can find that spontaneously. In other cases, though, we already know the gene because the nematode scientist found genes for programmed cell death in nematodes, and we're maybe studying mice, and we're like, I want to study that gene because I can do a blast search and I can find that there's a gene in mice that's really similar to what the scientists were calling SAD9 and what we're now calling caspase. And so, you know, I already see the gene I want to study, and I don't want to do a big genetic screen and look. So I'm going to build a null allele in a test tube and insert that into a mouse and make a transgenic mouse and make a knockout like that. And that's in contrast to a knockdown, so we talked about that in class, right? And those are just tools for researchers to learn, and that's how a big part of how developmental biology has roared forward in the last 50 years. There's a lot of genetics, so we got a little bit of that going with that. And we had a fourth quiz. What was that quiz about? I don't remember. Does anybody remember? We had a fourth quiz about something. And so that topic too. And then, but anyway, nematodes, and then we just were doing this ranging overview of the first chapter where we talked and introduced more about muscle, not just muscle differentiation, but genes. And then in the first chapter it says, well, these signaling factors, because we learned the rules of all life on Earth is, well, we're going to make a multicellular plant or animal, and it starts out, well, the cell is just a fertilized cell, and it's got to become different. And there's just these two ways of becoming different: unequal cell division or cell signaling. And unequal cell division, we have one example of that. That's the stem cells, the satellite muscle stem cells. They divide unequally throughout our lives to make precursors and maintain stem cells. And in every developing animal, there are examples of that unequal cell division, and it's called mosaic development. And that's happening also in the early frog during mitotic cleavage. That's a bunch of mosaic development, unequal cell division, because mom has loaded maternal factors asymmetrically, unequally distributed in the cytoplasm. Boom, 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 boom. And we end up with cells that are going to express glucocorticoid and become the dorsal lip of the elastopore, because of how we learned about that. We also are going to have cells in the vaginal pole that are going to release signaling factors like nodal and VEG1 and set up the belt of mesoderm. And so, we have some synthesis a little bit to pull, and studying to pull those examples together, because I could say, what are the two ways a cell could become different, and what are the names, and what do they mean, and then give two examples. And so for mosaic development, you'd be like, well, the muscle-like stem cells are what we learned about, and also early mitotic cleavage in frogs. Those are two examples of mosaic development. Again, there are examples in every animal, but those are ones we've learned so far. Then, in contrast, the only other way a cell can become different from the other, which is called general patterning, and our book is about patterning. There's just one chapter about growth, the other is about pattern, all the rest is about patterning. And the way they become different through signaling is the signaling factors that are produced from a source cell, and then a receptor has a target, a target cell has a receptor. And we initially learned about estrogen, so we could answer some questions about estrogen and estrogen signaling like that. And we've talked about that because that you inherited all the genes you need from your parents, and in some cases there's a big delay in using those genes. In the case of estrogen, the delay is until puberty, right? And I gave that example of my daughter Lena, who got her so-called 12-year molars and immediately got a cavity in them, because there's a big delay in that. And that's something we want to learn about. And a lot of that story can be started by talking about the relationship between these signaling factors, like hormones or other paracrine factors, and how they regulate the gene expression. And you can think of the DNA that we inherit as like an orchestral score, and then it's going to get performed. And you have these various delays and orchestration of the timing. And we have some examples where we can think about a sort of, here's a solo from like the violin. An example of a solo would be, MyoD. MyoD can turn a precursor cell into a muscle cell. So that's kind of like a dramatic example. More often, it's not a solo. It's the violins and the cellos and the trombones playing together. And so we try in IntroBio to provide examples of just like the solo pathway. So you can see, I see it. That's what our frog handout is. We pick genes, boom, 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 along that pathway. There are more, because it's a more complex, integrated musical performance than just here's a solo and there's going to be another solo and another solo. So that's going on. And we wanted to talk about signaling factors. So we had the Wnt drawing assignment and introduced that big concept, and we could give probably a 5 or 10 point question of like draw and label the Wnt signaling pathway like that. And in learning about the different proteins, most of the proteins are going to fall in our whole course into, is this a transcription factor? If so, it's going to do its job in the nucleus. Or is this a receptor? If so, there are these very unusual receptors called nuclear receptors and estrogen receptors, an example, that are in the nucleus and are transcription factors. But much more commonly, signaling factors, community cells, do not go into the target cell. They bind to a receptor on the surface. So we have, transcription factors, and then and signaling factors, which do not typically go into the cell. So then we say, or receptors, or components of the signal transduction pathway. And so we talked about estrogen receptor initially to give an example of the delay in development that occurs, but then to give a kind of a direct, brief example of a signaling factor that's special. The estrogen receptor, estrogen can go right into the target cell, right into the nucleus, and the receptor is a transcription factor. So that's a sweet example of a set of receptors called nuclear receptors, and they're super hugely important in our health. Estrogen receptor, testosterone receptor, and so forth. Another thing that popped up that students kind of struggled to understand is these transcription factors have genes they regulate, and we call those just in general target genes, right? So back to the muscles, MyoD has a set of target genes. We just learned one of them, but it activates other genes as well. Anytime a transcription factor is made, there are many thousands of those molecules in a cell, and they're ready to bind to any target gene. And so really, typically, a transcription factor has multiple target genes. We learned about estrogen receptor in part to appreciate that. The changes that occur during puberty are dramatic and throughout our body. And estrogen is doing that, or testosterone. Not just in our bodies, but all vertebrates have those as the sex differentiating hormone. And they make just dramatic changes, right? Like that, okay? So lots of target genes for the estrogen receptor and estrogen, right? And that's, generally the case. And so sometimes we just call the target genes. Like the Wnt signaling pathway has target genes, and those are also called target genes of beta-catenin, because that's the transcription factor that does the job that Wnt is regulating. And so really want to dwell on that, and then, along the way, we are also into other pieces of Chapter 1, and part of Chapter 1 is to talk about the vertebrate phylotypic stage and learn all those terms and the concepts of how vertebrate development starts from very different starting points because of the different eggs, shapes, and sizes in the variety of vertebrates there. But it all converges on a common ancestral form called the phylotypic stage, and then from the phylotypic stage, the vertebrates diversify. And so then we had the assignment, build this chick embryo at a vertebrate phylotypic stage. Why the chick? Well, who cares about chickens, really, right? But it turns out you and I look just like that, really just like that, at that early stage of development. About 1 month into human pregnancy is when we have that vertebrate phylotypic stage reached. So that's a big deal. And other types of invertebrates have a phylotypic stage too. There's a phylotypic stage for certain sets of insects and stuff like that. So it's a common feature of development of animals on our planet. And so those are lots of them, and I probably missed one or two. But it's all it's trying to stack it up to this, where we're picking up pieces of both that early descriptive embryology of what can you see. And sometimes that'll be the question on the exam. It'll be describe what you could see under a microscope for early frog development. And if we ask that, you want to describe that. And I'm not asking in that question anything about beta-catenin or goosecoid or cortical rotation. I'm talking about just what are those things. Or the question could be describe early frog development at a molecular level. And then I'm saying recreate this entire handout for 20 points. And another thing to mention about that is it's challenging to grade a 20-point question, because a student can do a great job and not name every single one, It can be like, that's great, you really have a solid, understanding of this. So I'm going to give you 20 points. And then another student can have somewhat different, but also really solid, right? Because, maybe one of you doesn't mention this, and then one of you doesn't mention that. So it's hard for me to grade, and I just go for it, because I think it's a really good way to learn this stuff, both to learn how to learn and also to learn the material. And so I just give it my best effort grading. Oh, and I should mention this too, because I haven't mentioned this before. When I grade exams, I grade one question at a time. So I grade the first question all across from the class like that. And I grade anonymously like that. So that's sometimes important for the student to be aware. I'm not just reading through your exam, and so I'm not aware if you've used this example, in a previous answer. I'm not looking, who you are or what else you've written on the exam. And so sometimes students will be like, I'm not going to explain this because I already did in a previous question. The last question I kind of told this and I don't have to do it again. That's not how I grade like that. And I'll just mention that anybody who grades differently than that is just incredibly biased. And you should just oppose that. You should check with your instructors. Because if anybody grades a full exam at one time, it's horribly biased. Because I'm reading and I'm like, great job, Bryce, great, right? And then Bryce gives a weak answer. And I'm like, well, Bryce is doing great. He probably knows that. Or, Violetta. Oh, Violetta's not doing too well, so I bet she's not going to get this one. It's a hard question. Yeah, it's just, and I should name the name, but even if you don't know the name, if you're just reading through somebody's stuff and you're grading, just get on a roll. You're like points, or points off. It's just ridiculous to grade anyway other than across that. The next thing to say about it is the way I grade stuff is I look at the answers students are giving and I base my grading on that, because sometimes there'll be something I really want you to answer as part of the answer, and none of the students have answered it, right? And I must have not explained it very well. So, that's on me, like that, right? So let's say it's a long question and nobody mentions Goosecoid. And I'm like, shoot, I called the handout Goosecoid. I really want to see that. But nobody mentions it, right? So that's important too, right? And of course, this idea of grading anonymously is just crazy important, right? Because I know some of you, and I'm like, oh yeah, Bryce is smart, like that. I've had Bryce in other courses, and I haven't, but you know what I mean, right? So be aware of that. Each question is new, just you're going to answer that, and it's probably not a big deal, but just don't think I've already answered that. Answer it again. And you can use the same example if it pops up. Just use the same example. I used it there for an ortholog. Now I'm going to use it here. And that that's fine. Yep. Okay. So that's some of that. Getting ready for that. Again, look at that first exam to get a feeling for the expectation, because it's it's pretty surprising to be able to spit out 20 points. Yes.
So when you're going through that review and just talking about, for example, the signaling factors in the estrogen case, and so just thinking back to it, so we have like the receptors, but then we also have like repressor molecules at some points, and you're talking about the delay. One analogy you gave just a moment ago actually confused me a little bit more. You said like, imagine as if it's like, a score, right, and different parts, but that conflicts with this kind of understanding of these things, at least in my mind. Maybe a misunderstanding is like, this analogy of like an origami folding plan and these things as tools, because that makes it seem to me a bit like it's, those kind of conceptually conflict in my mind, like that analogy you gave.
What was the analogy about the score thing?
You said like a musical, like, so like different parts coming in different points, and that to me just created some confusion because it conflicted with like thinking of these things as tools and because it sounded more like that, incorrect idea of thinking of things as like a blueprint, and so it kind of conflicted with that, and that analogy gave me some conceptual confusion.
Yeah. So, so I don't know if this will clarify it very well, but something that what I would want to mention, I think, in the context of that, is that developmental biologists say that development is robust. So that's a common term. And what that means is that it succeeds really often. And so what that means is that there are sort of checks and balances and ways to make adjustments if some step didn't work very well. And there's evolutionary pressure for that. And that's why there will be not just one factor that's getting things done, but there'll be multiple factors that are actually playing sort of partially redundant tasks to get things done. And I know that's not directly hitting the nail on the head of your question. But in that sense, we bring in the idea that the performers actually have some flexibility, so they're not just rigidly robotically playing the score. They are in, you know, classic thing, but in development, they're actually doing this more dynamic, kind of like a jamming thing, rather some improvised thing where they're getting to the goal collectively, and so that's what is evolving, is those coordinated things, and that's just kind of what popped into my head with response to your question. I know that's not directly addressing it. And so, you know, you said, yeah, there's this, you know, there's kind of the analogy of a musical score. And one of the weaknesses of that is that a musical score gets played out in a very rigid way, right? The director is up there going, okay, now. And realistically, it's more of this organic, fluid, improvised, or partially improvised thing, because it can get kind of bumped by different, like a player will fail to come in on time, and so other people will cover for them. Or there might be an environmental factor, like, oh, the lights in the auditorium turned off for a moment, and we got to keep playing, you know, like that. So we probably can talk more about that, because it's not a specific thing that would bring
Right,
up,
yeah,
you know,
okay.
answers on the
Yeah,
exam,
fine, later.
but more of a feeling.
Can I ask
Yep,
one other
yeah,
more specific,
absolutely.
more relevant? So yesterday, and I'm gonna refer to this little
Oh,
figure,
yep,
sorry,
yep.
it's what you were drawing, and you're talking about the development of like the belt buckle versus the mesoderm. Sorry
Yeah.
if that's a terrible drawing. It was.
No, it's, yeah, great. No, it's just what we were drawing, you know,
Yes.
that we're gonna make a belt.
Can we briefly review kind of like
Yep.
how that goes again?
Yeah, so we saw, let me show you this figure, because it's a very useful visual. I gotta drive around, you know, because I kept some of these slides up and removed some like that. That one. So these are two genes whose names we're learning that are involved in specifying mesoderm. So see how brachyury is a transcription factor that is going to say to all the cells, you're a part of a belt. And then we have a special belt region, mesoderm region, that you're going to be the buckle. And again, I'm the only person in the world that talks about a belt and a belt buckle, but you can see it's a fine analogy, because a belt buckle is part of the belt. And so you're going to be a belt if you express brachyury. And brachyury is a transcription factor that's going to activate target genes that are mesoderm genes. Then goosecoid here is going to be the gene we learned about in the buckle that makes it a buckle. And so the handout is really organized like this, where steps 2 and 3 are really telling about signals that are going to be floating up from here to say, be a belt. Everybody along here, be a belt. There's complexity in 2 and 3 because it's also trying to add this idea that mom makes these maternal factors, and the maternal factors have to be made in mom in the ovary, and then they have to pause until fertilization. So mom builds tricks in. For example, lots of maternal factors that will be functional proteins in the early embryo are loaded as just the mRNA. So that's true for veg T. Mom says, I gotta put these in to the maturing egg in my ovary before it's ovulated, before fertilization. And I have to build in automatic pauses. So there are a bunch of maternal factors that are loaded as as mRNA, and the poly A tails are too short for the ribosomes to transform into protein until after fertilization, and fertilization causes kind of the resumption of development. Development is already occurring, you could say, in mom's ovary as the egg matures. We're going to restart it. So there is that addition of those details in 2 and 3 on here with veg D and veg 1 and like that, okay? But the bottom line is these guys are going to become cells during mitotic cleavage, and then they're going to start expressing genes, zygotic genes, and some will be signaling factors, and maternal factors and zygotic factors will float out. And these cells have receptors, and they'll receive those, and they'll respond, and they'll respond by making brachyury. That's the end stage goal of 2 and 3. Then 1 and 4, and remember it's 1 and 4 because there is this important inevitable delay. Something's going to happen shortly after fertilization. We're going to have transcription factors ready to go, but then during mitotic cleavage, there is no G1 or G2 in the cell cycle. It's just S phase, M phase, S phase, M phase, because we're trying to slice up this pie into cells. So we get some stuff done on 1, and then at 4, at the end of mitotic cleavage, when we get to express zygotic genes, that's what's shown here, is zygotic gene expression. And so in 1, we explain how we set this area up is going to be special. So even in the single cell fertilized egg zygote, we have this movement of some disheveled up here, and it's going to become a special region because disheveled is going to protect the beta-catenin from destruction, degradation here. And so we're going to have a special transcription factor, and the special transcription factor is waiting to do its job. It was protected in the early embryo, then it has to wait until late blastula, and then it gets to do its job. And it binds to the enhancer of Siamoas, and we get this little pathway that's very common and is remindful of the MyoD activating myogenin, and it's a little cascade of gene expression that gets activated here. And we want this to be a crisp switch. All the cells have Siamoas, but we don't want Siamoas expressed anywhere except there. So that's part of also what we're learning about is just these examples of crisp molecular switches. Siamoas is not going to get expressed because TCF is repressing it, is actively keeping Siamoas off, and only here is the beta-catenin stabilized and binds to the back as a co-activator on the back of TCF, overcomes the repression to actively express Siamoas. And this is happening in the late blastula, just before the start of gastrulation, and it's happening in what's going to be the dorsal lip of the blastopore. And we saw that, famously in chapter 1, you can take these cells and transplant them to the tummy of a host and get a conjoined twin formed there. Because these cells are going to express signaling factors that tell other cells what to do. These cells are both target cells, become a belt, but they're also source cells, become special regions like that. Yeah?
I had a question about, it's related to the Wnt pathway, but it at the core is the idea of the source cell and the target cell. A source cell, I'm assuming it's variation, but for the most part, can we assume that the molecule they're releasing could be released constantly and it's at the receptor side where that receptor is either expressed or not expressed to prevent it from going in? Because I was a little confused with Wnt.
Yeah.
Like, what's keeping
Yeah.
Wnt from going in all the time?
Right. Yeah. So, when that question is about, the organization of the general idea of a source cell and a target cell. And we want that switch to be tightly regulated. So in the case of estrogen, we can think about production of the hormone. hormone's not being produced in the child, and now it's being produced. The child has the receptor. In that case, very dramatically and significantly, the receptor is functioning in the child to repress target genes, like that. So it's there, and it's doing a job. Very, very surprising, but a molecular switch. So in that case, the story is all about estrogen production, okay? As we're learning. Now, the next thing is, there are plenty of cells that do not respond to estrogen during puberty. Our eyes, nothing happens to our eyes. They probably do not have the receptor, right? Like that. So, you know, it's a pathway, and it can be regulated in multiple steps. Well, is it in the source cell and making the signaling molecule, or is it in the target cell and having the receptor or components of the signal transduction pathway, or the cellular response like that? Yep. Yep. Yeah, and what we're talking about is a gigantic show, and we're carefully introducing the principles and using details for some of the principles. Like this, you should, hang out with your family or friends or just on a bus and just start swearing about how frustrating it is for you in your life to have to learn this about frogs. Because who cares? Does our body work this way? No. We don't have anything like that, right? The key thing is from this you start to get a feeling for what is this thing that's happening. And there's gotta be a lot to the story, because we're the product. And every other animal on our planet is the product, every generation, right? So it is the thing, where you're like, dang it, this seems like a waste of time. But there's also nothing else that's really worth studying, like that, right? Like certainly not Latin or Greek, sorry, but like that, right? It's the coolest thing. Yeah, because scientists know so much about it, and it's in the news all the time. And it's not well described in the news, but once you have some developmental biology, you can Google it and start diving into it and be like, oh my God, this is fantastic. Yeah, I'll just mention this side story, that kind of signaling, that happens in our bodies now, and it's really significant. So like last Monday, the scientists won the Nobel Prize in Medicine for how the immune system is regulated to kind of keep it from attacking us. And that's a bunch of cell signaling. The cells are talking to each other. Signaling factors in an immunology course are called cytokines and chemokines. But it's the same idea. There's a source cell that's communicating to the target cell, and they're coordinating their efforts. So it's just, yeah. But anyway, okay, other questions people have? Yes?
When you're done with the exam, do you post an answer key video for it?
Yeah, I always make answer key video. Yep. Yes. Yeah. And so I'll do that. And so when I grade the exam, a lot of times I'm just, marking points off. I'll write okay sometimes on an exam because it's like, okay, that's, I really don't like that answer, but it's official enough and to give it credit. But when I write okay, I mean, especially look at the answer key video, because I gave you points, but there's a much, better answer. And so we're trying to keep learning. Even if you get all the points, you're trying to keep learning. So, everybody will do the answer key. And, I'll probably, depending on how things go, I'll probably have some kind of follow-up assignment, like go through your exam in the answer key video and answer, 10 questions you got wrong or something like that, because it's really significant to get that feedback, yeah? And I do it partly because I want to write the same thing, on 8 of these exams, because they're answering it wrong in the same way. They misunderstood it. And everybody would help, for me to anonymously explain, this is not it. Because there's some people that didn't put that down, but they're like, oh, I didn't realize that was, part of, that could be part of the wrong thing, because I was kind of wondering about that. Yeah? Yeah, so I definitely do that, yes. Yep. Other questions people have? And we're not in a hurry, we're doing great, but I'll start talking about a little bit more that we can add to this, some stuff that's from chapter 3, but especially chapter 4. We're never gonna look at chapter 4. It won't help you on our exam to look at chapter 4. You know, what I said is just don't bother with chapter 4, okay? And if something else pops up in your mind, you know, you're thinking about right now at the exam, you know, just interrupt. And I'm just reminded that I used to always say, does anybody have any questions or comments? I don't know why, it seems random. But one time, I had a student who's long gone, her name was Haley, and Haley would have comments and would stand up to say them. You know, she's just really great. And I'm just like, yeah. I talked to a student yesterday on Zoom who graduated from here in 2005. And he was like that too, just a very interesting, unique person. And he's out there having a lot of success in working in a science company, a research company in New York City. And he talks to me every once in a while. And it's fascinating, and I know each of you are unique in your own unique and different way, and it's just, yeah. So anyway, if you have questions or comments, and if you want to stand to say them, you know, just go for them. You don't even have to raise your hand. You can just stand up, and I'll be like, oh, what's Blair want? Like that, right? All right, so let me show you this, because it would be good for us to get a chance to talk about this some. And it's small packages, right? We got the big packages, but here's something, and that is, you know, we did this assignment, and I said, well, that's what a mammal embryo looks like at the phylogenetic stage. So let's just talk about mammals a little bit. It's the same thing, but what is that? How does that fit in to our real human pregnancy? So that's going to happen here. You can already see there is a picture of it. And instead of looking like a disc, like a flat disc, it's the same thing, but just imagine the disc is curved up into a bowl, a flat bowl, right? So it's happening there, it's happening in the uterus, and here is the little embryo at about the phylogenetic stage that's been implanted in the uterine wall. It is going to make that nourishment and gas exchange connection with the mom to the placenta. So let's just get to that stage and talk a little bit about a couple of terms that we need to learn that will be on the exam, okay? So here's a mammalian pregnancy, you know, very different than non-mammalian birth, because ovulation leads to the release of a very little egg. There's no need for any yolk, because we're not going to build a little chicken or a little tadpole that's got to be able to get out of the, you know, and start eating, because the embryo is going to develop to, you know, pretty long developmental process in a mammal to, a baby that gets born like that, or a pup, you know, or a kitty like that, right? So we have ovulation, and then the egg starts to move like this. And fertilization is going to take place here as it just is floating down toward the uterus. And so it's showing here. And it can take place over a couple of days, and it occurs here. And then we don't have mitonic cleavage. We're not in a hurry to divide up a big fertilized egg. The cells divide slowly, and we have a 2-cell embryo and a 4-cell embryo, and so forth. And we're going to get to, in about a week, plus or minus a couple days, we'll get here and we'll have implantation. So prior to implantation, this is called a pre-implantation embryo. And it's very different than a frog or any other non-mammalian embryo. For example, it appears that there are no maternal factors in a human or a mammalian egg. And so just a dramatic change, right? We talked about that, right? That, yeah, all vertebrates are going to develop to a phylotypic stage, common, similar stage. They start at very different places. And, you know, we emphasize big egg with lots of nutrients that's going to get divided really fast, and also dividing up these maternal factors to have the cells starting to become different. Here, in the pre-implantation stage, which lasts a week to 10 days, all the cells are equivalent. There's no maternal factors, there's no mosaic development, no unequal cell division, and they're just given a special name, and they're called embryonic stem cells. And embryonic stem cells are famous because you've heard about them, and so we want to be aware of that, right? That, again, we learned about the chicken embryo, and at this stage, the human and mammalian embryos look like a chicken embryo, but earlier, they're very different. So let's just look at that and draw that. So we've learned that, and you've heard, like, this is an example where, you've heard about embryonic stem cells before, and, you build some kind of knowledge based on how they pop up in the news. But, so we learned about stem cells like the muscle satellite stem cells, and they're dividing, and they divide like this. They divide asymmetrically. They become unequal cell division. The stem cells are said to be self-renewing because we never lose them. We don't need more muscle satellite stem cells because they're self-renewing. They're good throughout our whole life, no matter how many injuries or growth or working out we do. Yep. A person, if they are not exercising, their muscles atrophy. Like that. That happens if a person has their arm in a cast. Their arm muscles atrophy, they're not being used. But as soon as they get out of the cast, well, they start moving again. They need to build that muscle up, and that involves a lot of muscle satellite stem cells. So we have that example. So here's how you would draw an embryonic stem cell. The early pre-implantation embryo of all mammals looks like that. The cells are dividing symmetrically, and that's literally the terminology. This is asymmetric cell division, unequal, and this is symmetric. And they're equivalent. And that's why they're embryonic stem cells. And interesting, because you see that if you were a scientist and you said, I want to study some stem cells, and you got these muscle satellite stem cells, you got them in a dish, you're never going to have more than you started with. But if you take an early mammalian embryo, you can get it in a dish and split the cells up, you can just expand those indefinitely. You could get trillions of cells by just growing in a dish and keeping them separate so they're not on their way to forming an embryo. Yeah, and so that's why embryonic stem cells are popular. Another thing to say is, are these ever acquired through a abortion? No, because nobody has elective abortion in their first week of pregnancy, and they're gone. By the time the embryo starts implanting, those embryonic stem cells start to become different from each other by self-signaling, unequal cell division, and there's no such thing as an embryonic stem cell. Pretty soon we see these stem cells appearing like the muscle satellite stem cells. Those are not embryonic stem cells. See, there's a big difference. And so where do scientists ever get an embryonic stem cell? And the answer is from in vitro fertilization. And when folks go through in vitro fertilization, each month a woman ovulates really typically one egg. And in vitro fertilization is inefficient. So if a couple is going through in vitro fertilization, the doctor gives the woman drugs that cause her to superovulate in 1 month and release 6 or 7 or 8 or 9 eggs. And they collect them all, they fertilize them all in the dish because it's expensive, and then they just freeze some of them and they implant some, because that implantation process in in vitro fertilization doesn't work all the time. So they're like, let's have some in reserve. And you probably have heard that idea that, you know, somebody who goes through in vitro fertilization, they implant some, and that's not very efficient. So they'll implant 3, and the couple might have 2 babies, and they'll be fraternal twins, and they'll be like, our family's done. And they're like, well, you have some more frozen embryos. You want to donate those to science? And that's what they're donating, a pre-implantation, early embryo, and scientists can grow, and you don't need many of those to just grow millions and millions of cells and, you know, share them and study them, okay? So a little bit about, you know, the very unique feature. That causes this phenomenon, so let's just look at that. You don't have to scramble to write this down, it's not in our textbook, but this is just showing identical twins. And when we were in our mom's tummy, I've described this before, we are producing our bodies, that's called embryonic cells, and we're also making cells, our cells, to work with our mom's cells and make the placenta, the amniotic sac, and other what are called extra embryonic cells. And so that's just shown here, and what's shown here is if you know some identical twins, you might have heard them say, oh, we're identical twins, and we were in the same amniotic sac in our mom. And that's just based on the timing in that first week of when the single embryo happened to just get split in 2 or 3. And so you can see here that you have these different situations. Here's where the identical twins are sharing the same amniotic sac, and it's because of the timing when they split, when they happen to split. Or later, they can be in separate embryonic sacs, but share the same placenta. Or later, they can share, have separate amniotic sacs and 2 placentas, like that. So those are just details, natural details of the identical twinning process that occurs in mammals, and identical twins just don't form in other animals because they don't have those embryonic stem cells. The embryonic stem cells are identical. So you can think about that, right? You can just think about, like, it could just be a 2-celled embryo, and if it divides, you're going to get 2 babies out of it. They are able to regulate their situation. They're like, oh, I can make a whole embryo, I will. I can make all the cells, you don't need it, I will. You know, like that, okay? So that's an important thing to talk about. And so then we get this implantation, and we start to get the development of the extra embryonic cells, the embryo, and the connection to the lining of the uterus, and eventually, you know, a umbilical cord, like that. And so there's the embryo, and it's just developing the same way we learned about the chick, like that. So in this process, in that early development, we should just look at this and learn 3 terms. And again, this is not from your textbook. And what's going on here is we want to emphasize that in the frog, when we talked about the frog, we made a big ball of cells, a hollow ball of cells called a blastula. The chick did not make a hollow ball of cells because of the huge yolk. The chick made a flat disc, coin, frisbee-shaped embryo, and that's called a blastodisc. Some people call it a blastomere, but a blastodisc is a great name. So does the chicken blastodisc, does that have a blastopore, a dent? No, it has a primitive streak. So we're distinguishing those in this early vertebrate development. Frogs and amphibians make a blastula. Chickens make a blastodisc. These guys, mammals, make a blastocyst. And that's just good because students often confuse that. And it's important to appreciate we're headed toward a phylotypic stage, but at the beginning, it's really different stories. So a blastocyst is what we want to learn. So that's a term, and then this is the next, and that is that the blastocyst is a solid ball of cells, and And right about the time of implantation, we start to see the cells become different from each other. And our book has figures about that in the textbook. And so just to show you this, again, you don't need to look at it, but here it's showing right about the time of implantation, we start to see a mammalian embryo's cells start to become different, the first differences. And they can follow their fate. Now we can make a fate map. If you make a fate map of a really early pre-implantation embryo, you just tag the whole embryo, you know, because every cell can make the whole organism like that. Here, we start to see some differences, right? And the early differences are really which cells are going to make the embryo and which cells are going to make that extra embryonic cells. Because that, we gotta make a plan, right, like that, right? And so the terms we want to learn are inner cell mass and then this trophectoderm, okay? And that's it. And so just to get, that's a visual of the three terms we should learn. Blastocyst, that's what's coming along down the fallopian tubes. It's going to implant. Right about the time you implant, we start to see these differences. And here, trophoblast, or this is, people call this the trophectoderm. So you just learned, you know, trophoblast is great. And so let's just look at this. We have the inner cell mass and the outer layer of the trophoblast, trophectoderm. And that trophectoderm is fated to just make extra embryonic tissues. The inner cell mass is going to make the embryo plus extra embryonic tissues. So just to show you that, because that's the time when we're not talking about embryonic stem cells anymore. At the time of implantation, the embryo is starting to have cells that are becoming different from each other, and they cannot make the whole embryo. We know what's happening. All along the way, there is genomic equivalence. The cell has all the genes it needs, and it's starting to have differences in gene expression. The trophoblast is going to be expressing some different genes than the inner cell mass. And that's how it's going to have a different fate, right? This is you applying to college, right? Just very early and just starting to have a fate that's different. And that's why we want to learn about it, is just to make that, kind of really appreciate the unusual feature of the early mammalian pre-implantation embryo. All the cells are equivalent. There are no maternal factors. Okay? And so that's that. And that's it. You know, if we could learn those three terms. Now again, those are probably like fill in the blank, right? Right? You know, they're just little parts. So we're not learning huge things today, not asking you to go out and scramble and learn a lot more that we haven't already covered. But it's very good for us to see that. And I just want to add just a little bit more. A term you don't have to learn is, lots of people call this early blastocyst of morula. That's actually the more common name. Like if the New York Times is going to write an article about embryonic stem cells, they'll probably mention the morula, because the morula is another name really for that early pre-implantation embryo. And so there's kind of a confusing overlap. So don't worry about morula, I'm just saying it. We have a great term, because you can hear blastocyst is different. That's mammals. Blastodisc is chickens and reptiles and birds, and then blastula is amphibians, right? So distinguish those three to appreciate they're different, and they're going to be coming closer together like that, okay? So another thing just to show you is this figure, and we're not going to talk about it today, but we're going to talk about it when we talk about stem cells in about chapter 6. And what's going on here is an experiment like that. And scientists are like, oh my goodness, so you're telling me that I could take a pre-implantation mammalian embryo and split it in two and get 2 embryos out of it. Well, what if we do this experiment? What if we take 2 pre-implantation embryos and we just join them together? What's going to happen? And the answer is that although that's not natural, the mammalian embryo adjusts and makes one organism. And what they did here to try to give you a feeling for that is they took an early pre-mutation embryo from white furred parents and an early one from brown furred parents, and they just joined the pre-mutation embryo together and put it in so that, you know, let the early development happen in vitro fertilization. Joined those together and put those in, planted those in her womb. And this is the baby mouse that's born. And it's got patches of brown fur and white fur because it has 4 parents. The DNA between the strains, white fur, brown fur, the DNA does not mix. They're different cells like that. And we're going to learn about that later in our course. And for now, it's just kind of a novelty. But you'll see later on, it becomes kind of a cool experimental tool for this reason. If you take this mouse and you look at the other baby mice that are born, they each have a unique pattern of brown and white fur, just random. Some have very little brown fur, some have a lot. It's just their chance thing of what the cells have to contribute. If you take each one of those and you raise it up and you breed it, its sperm or egg are only from one of the sets of parents. So let's say this mouse grows up and it's just that generation that's affected. All its babies are going to be, we'll say, brown furred babies. Yeah, and so it's just a curious feature of that generation. But it's nice to see at this point, because you can say, yeah, that makes sense, that would happen. And that's a mammal doing that, you know? And they regulate or adjust the development to make it normal. Yep, okay. So that's the last thing we want to mention about these. And we'll come back to this exact figure, you know, in about a month, because there's an experiment that scientists do with them that is very useful. Okay, so that's that. Now I just put in some, you know, things like this because we want to make sure we're solid on this. And if anybody was going to ask any questions about that, you know, we have these figures here. And I want to show you these because they're from, I think they're from chapter 3, and we've talked about them, but just so as you're reading through chapter 3, you see, oh yeah, we talked about it. So this is the early experiment where they're like, that belt buckle gets set up when the disheveled gets moved from the ventral fold to this region. And so even though this is just a few cell embryo, it already has the active disheveled. And I mentioned last time the name Nuku. It's just the name of a scientist who was in Holland, where they have unusual names, you know. And the scientist just thought, in Germany, the scientist transplanted the dorsal lip of the blastopore cells and twinned the embryo, right? And Newcoop just thought, look, how early could I do that? And the answer is very early. As soon as the disheveled gets moved up, that cytoplasm becomes fancy, ready to make a buckle, because that has that disheveled in it. And Newcoop didn't know that, because it was probably 70 years ago that Newcoop did experiments. So Newcoop didn't know that, but just got a little needle, pulled out some of that cytoplasm, and transplanted that into a host tummy area in the early embryo and got twins. So the experiment is just showing that, you know, just showing that, yep, what we talked about in our handout, that gets set up really early. And even before, you know, we have the dent, the blastopore. And that's going into the idea that, right, we get fertilization and some events happen pretty quickly, but then they have to wait for mitotic cleavage to get going, right? And this just shows, yep, there were events that were early and they were ready to go, but they were waiting, you know, to, you know, for the stabilized beta-catenin in that region to get to do its job, okay? So it's just a figure. And, you know, we don't talk very much about the experiments that our book talks about. You know, our book, this is an experiment, and it provides experimental evidence for, this story here. So just see that, that's what's going on, and they help you navigate it, right? And so here it's just showing the experiment, right, where they're transplanting that, what's called the new coop center, in honor of that guy, and they're getting a twinned embryo. And then here's an example, of that fate map, and I had that early fate map that shows that early on there is no mesoderm specified. Early on, the early embryo thinks, I'm just going to make epidermis and endoderm, the blue and the yellow, like that. And so this is just terminal evidence for that from the book. Again, the book doesn't explain it the way I did in class, where I made that special figure. But this just shows that you could do, right, we could do the experiments to make a fate map, right, tag it. We can also do this thing where we transplant it. This is that experiment, the test to ask for specification. What if you were a pre-dental student and we had you just live by yourself, so you didn't have any other influences? Do you know, is that what you're going to really, do you know that you're going to become a dentist? And that's called specification, where we don't transplant the cells, we actually move these cells just by themselves in a dish, sterile conditions, nutrients, and just see what they become. And early on, the early specification is, I know I'm going to become endoderm. I know I'm going to become ectoderm. And remember that early on, no mesoderm is specified. This belt is going to become specified as mesoderm. Early on, it actually thinks it's going to become ectoderm. And we have to tell it, no, we need some mesoderm. And it hasn't heard that yet. So specified means these cells in this belt region can still change their mind, but you have to leave them in the embryo so they get the signals to change their mind, like that, okay? Yep, so just a couple of figures from your book, because the book does that, and I don't really talk about those because it's some of the experimental evidence, and it's really good for science, just to see some of the evidence and think through the reasoning like that. Then I think the other piece I just wanted to follow up, so we're all square on this, is that we were talking, really up till here, and we haven't really talked very much about neurulation. You don't have to know very much about it, but that happens before the phylotypic stage. And so here is a picture from chapter 4 that I've drawn out before, and it shows the neural tube forming being induced by the notochord, and then it shows the appearance of those neural crest cells. And so we learned about that, and I'll just draw it again because we've seen it, and it's this story that is the story of neurulation, right? So in the frog, we get fertilization, mitotic cleavage forms a blastula, then gastrulation occurs, morphogenesis, we get the blastopora and the folding, and then neurulation. And neurulation and the neurula just means the neural tube is forming. And we have this situation here where we have the notochord, and these are just the overlying cells that are fated to be ectoderm. And they think they're just going to become skin. But the notochord are releasing signaling factors like this, and these cells are going to change their fate from thinking they're supposed to be skin, being specified to be skin, to becoming the central nervous system. And I'll just draw this as a little line. The way that happens is we get a dent like this and ridges forming above the notochord, and they fold up like this, and then they're going to pinch off, and we'll get the neural tube out of it. And the neural tube is hollow, and we talked before about how our adult spine and brain are hollow. They're filled with cerebral spinal fluid. That's happening here. This is the neural tube, and the neural tube is a hollow, like a garden hose, and the cells are the tube, and they're going to make the whole nervous system. And they're induced like that. And then I also want to draw in that the cells that are almost going to get in but are not in, they're on these neural ridges or neural folds, and they're going to remain here as part of the ectoderm, and those are the very unique to vertebrates neural crest cells that we learned about. So that's another thing, right? So, you know, when I said, oh, let's summarize everything, that's something I didn't mention, but yeah, we learned some about the, well, it's part of, I guess, the vertebrate phylotypic stage. It's a huge feature, hugely significant feature of the vertebrate phylotypic stage is, at that point, just along the dorsal midline are these stripes of neural crest cells, and they're going to migrate. So we talked before about how the belt is going to fold in, and the belt folds in by you guys just kind of jostling. Nobody's really like picking their desk up and moving it. You know, you just kind of push together, and the students that are in the front get pushed forward, and the students on the back, they get pushed backward. But nobody's actually crawling around like that, right? Neural crest cells are different, and that's called, they migrate. So that's a different term. They don't jostle, they actually move, like you leave the building, that's migration. And they migrate up to form the vertebrate face that gets zipped together. They migrate out to make the peripheral nervous system, and we'll learn more about that late in our course. And they migrate out to the surface of our body and any mammal body to form the pigmented cells. And they make some other components too, but sort of minor components. So we want to be able to like describe the neural crest cells and give examples of what they produce, you know, on our exam. So another kind of smaller piece, and from chapter 4, but we've talked about it, right? Okay, so that's like that. So just covering some of those things like that. Okay, so again, we're doing great. Oh, and I forgot my phone. What time is it?
10:43.
Ah, so we got to stop. Let me just say that, yeah, we're going to have our test, and, you know, I can, you know, you can hear from me and stuff like that. And also, I'll just say that, you know, there was a question about chapter 2. Yeah, no, we're not covering, we don't have time covering chapter 2. And I decided, and I'll make an announcement about this, but, you know, you're focused on the exam. After the exam, I decided we're going to actually read chapter 12 about plants first. You don't have to look at that. You don't have to look at, so we're going to run about chapter 12 plants, and then we'll do fruit flies. Yep?
I was wondering, because I know you mentioned that you weren't going to post like a key video for the old exam. Is there a reason why specifically? Because I think personally that would be really helpful.
Right. So yes, I agree. And so, so I've posted old exams at the start of my course, every course, forever. Because, well, how do you know what's coming, right? And it's not about knowing and using it as a study tool and being like, I got all those answers, I understand them. But instead, seeing the format and appreciating, oh my God, I see, he's really gonna ask those questions. He's not kidding, right? And I don't post an answer key because it is good for you to look and not just look briefly, but actually kind of dive into it and say, he could ask that question on you, you know, you should prepare the answer. And what I found is if I post the answer key, because it's an old exam, so I have the answer key, but if I post the answer key, then students don't pause on it at all. They're just like, God, I got that. It's going to be a hard exam. But, you know, it's good kind of practice to not have the answer key and not say, you know, am I going to give it out, you know, later, you know, like this weekend or something, you know, to check your answers? Because, you know, you kind of have to prepare the answer with, you know, confidence of like, God, if you ask that question, and, you know, you can answer it. So, yeah, so that's why, and I don't post old exams for other things. It's just the first one, just to see what's coming, you know, like that. And then, yeah, and I don't put the answer key up because I do want you to go, geez, ooh, I have to deal with this. You know what I mean? Like that. Rather than be like, oh, yeah, this is going to be hard, but whatever. Yeah, because, yeah, when you look at the old exam, you'll be like, God, that's a familiar question, and I don't know a very good answer to it yet, you know? Like that. So, yeah, that's what's going on with that. Yes, and again, we can keep communicating, you know, about that, because we got a week to go here. And, yeah, it's a bunch of stuff, but, you know.